university professor at the
something like that.
University of Texas, San Antonio. Most importantly, and which will be completely lost on most people here, he is a fellow Gaucho, so go Gauchos.
You're not a Gaucho, are you? Okay. Great. Thank you very much. And I'm glad that this sort of worked. Let's see. Great. So this is Babbitt's change of me talking about redox chemistry and the brain. He said that wasn't sexy. So it's also our approach of trying to really get at some new targets for Alzheimer's disease. And I think it's really great that this presentation follows a much broader holistic view of the disease, that it isn't just one thing that's changing, it's multiple things that are changing. Okay, so the work I'm going to present is collaboration, and I won't give credit to everybody, but people at UTSA doing advanced microscopy and mass spectrometry, my people I used to work with at Case Western, particularly Mark Smith and Larry Sayre, that both passed away after I left Cleveland, as well as notably the group in Warwick that we've been working with, a facility at Oxford University. So where did Alzheimer's disease start? Where did it go? Did he get it all wrong or right? Well, he was describing a middle-aged woman, August Dieter, and her brain, when she died, that she had plaques and tangles. In fact, this was the first description of the neurofibrillary tangles. The plaques have been noted before. In that description, he doesn't say that they're causative or anything else. There's just a description. She had a lot of other psychotic behavior that really doesn't fit classic definition of Alzheimer's disease. And it was thought to be an obscure disease because this is a disease of middle-aged people. Well, moved to the 1970s, and Tomlinson, Blessed, and Roth studied people at advanced age in a very consistent manner and found that they had similar changes in their brain to what Alzheimer described. And you move from a rare condition to a public health issue and major issue. So right now, there probably are over 50 million people in the world living with something like Alzheimer's disease description. And this number is growing. It's not that Alzheimer's disease, the incidence particularly, is growing. The risk at any age, what's growing is the population that live over 80. And that population is really very susceptible to disease. Some estimates are between 30 to 60% of people over the age of 80 have major dementia that would be classified as Alzheimer's disease. When I mention 30 to 80, it depends on your socioeconomic situation. Very relevant for all that multifactorial thing, genes, environment, education, et cetera. So and all of us know that the impact financially but socially on families is great. Well, you could say, geez, it's only been studied since 1970 and it was thought to be an obscure disease and not a lot of importance, but there have been hundreds of thousands of papers. When I estimated this, 130,000 papers on Alzheimer's disease. Actually, depending on what type of keywords you use, you can come out with 250,000 papers. And I estimated that there were probably about 60,000 investigators, if you look at people at least they're involved in a couple papers. So there's been 115 years, thousands of people, and a huge amount of money, especially in recent times. The NIH is spending over $4 billion a year in funding. The drug companies have spent over $40 billion developing various types of amyloid-related drugs. Yet the estimate that Jeff Cummings made is that 99.6% failure. And that was a few years ago, so it would probably be 99.8% failure currently because there'd be a little bit more tests. Well, has the focus really been not moving away from where Alzheimer's described? Alzheimer didn't say they were causative, but has it been with the genetics which showed that amyloid mutations in the amyloid pathway were associated with the disease? Has that been the problem? Or moving to tau the same way? And that's what I'm going to be talking about what role these proteins may play. Okay, so there's been a ton of targets. These are some of the 64 known targets for Alzheimer's drug discovery. So it hasn't all the work been on amyloid, but they've all been on single things, toxic compound or something else. So we took at this a way of looking at Alzheimer's disease that focused on oxidative stress. And some people here ask me about my original training, which is as a marine biologist. And that's what I studied when I was a marine biologist, was oxidative stress at fertilization, the beginning of life. So the classic definition of oxidative stress is the production of reactive oxygen in excess of protective mechanisms. I call it the rusty nail idea. And it's basically because chemists dominated this field. They view things as very non-dynamic. And you make something, you're young, everything's vital, and you're getting old and everything's going rusted and going to crap, okay? But that isn't really the way oxidative stress really works in living systems. Because what happens is when you have an insult and things become rusted or changed in terms of having an oxidative modification, the system tries to bring it back to equilibrium. Because there's a consequence in life of not being in equilibrium. It's called death. When your cell is in imbalance, it's going to go to apoptosis or some other pathway, and when your body isn't at the right balance, it's going to go downhill. Your body's always correcting itself, and it's why there's a robust sense of life. Okay? So what did we find out when we looked at oxidative damage? And it wasn't that we understood everything like day one. I'll tell you, I was pretty confused. And that was initially when we did our studies, we thought oxidative damage would be on the long-lived polymers like tangles and plaques, and they would be responsible for the insolubility that Dennis Salko described in the early 80s. When we looked at this more carefully, we actually saw oxidative damage was much more pervasive and wasn't particularly associated with the pathology. So here is sections through the brain, the hippocampus of somebody with Alzheimer's disease in an age-matched control. A and C and all the lower panels are somebody with Alzheimer's disease, and B and D is an age-matched control. And you see the difference is pretty much black and white, but also all the neurons look the same. This is oxidative damage to nucleic acids, RNA, mostly ribosomal RNA, but not completely limited to it. Okay, so why this was a surprising result was that it wasn't limited to the pathology at all. Even down in F, you'll see that there's a neurofibrillary tangle there, marked by the little arrows, and that neuron has a little less staining. I'll come back to that later. It's 50% less staining. So it's actually unrelated to the pathology. So if I put this in this drawing, which doesn't really exist except for the cartoon I made of it, we looked at what happens, what is the time course of the changes in oxidation. So if we look at that, we have a normal neuron on the extreme left, and the normal neuron actually only occurs in people under 40 years old, but that won't be part of my talk now. There's already most of these changes already when people are over 40, to a lesser extent though. And when people have Alzheimer's disease, we found all of these stages, even the extracellular neurons, the ENFT, the INFT, the pre-tau change neurons, as well as neurons that had nothing abnormal about them. They all had oxidative damage, and they had oxidative damage involving sugars, proteins, lipid peroxidation, and nucleic acid oxidation. And why this perplexed us, because the large range of oxidative damages I'll talk about later is probably caused by Fenton reactions that work very, very close to the site where they're produced. So that meant within the neuronal cytoplasm, if a normal appearing neuron in somebody with Alzheimer's disease, there's an abnormality that had not been described. So we looked at particularly mitochondria, because they're the primary site. All the oxygen you breathe is going to your mitochondria, or the vast majority, and being used to metabolize food. We also looked at redox metals. They are the ones that catalyze your food even when it's normally metabolized, as well as the metabolized oxidative stress and the role of amyloid. Okay. So this is a schematic of one version of the Fenton reaction, and this could be what's causing the oxidative damage. There are metal-independent pathways that cause oxidative damage, but everything points to metals being a key feature of it in general oxidation, as well as in this case. And you can see here that there's a strong interaction between iron and copper, changing their redox state, and playing a role in converting basically hydrogen peroxide to hydroxyl radical, which is capable of oxidizing any compound. In fact, it oxidizes water even. So we did some material initially, developed a technique for being able to look at redox activity, which we call, in this case, iron localization, redox iron. But in fact, we're less sure that it's completely iron now, because it was based on displaying a particular ability to change the redox state of iron. And we found there was a big change between Alzheimer's disease and control, and that change occurred during mild cognitive impairment, during the transition between Alzheimer and control. And this led to the following idea, hypothesis of how oxidative damage might occur in the disease. And that is that mitochondria, and I'll turn to why we thought mitochondria would be at the center of this, that mitochondria may play a critical role in changing the metallobiology of the cell. Not increasing the amount of metal, but in changing its redox state. And mitochondria, when they have problems, both release superoxide, which is the O2 minus, and the O2 minus easily gets converted by superoxide dismutase to hydrogen peroxide, and that interacting with metals by the Fenton chemistry I just showed would lead to hydroxyl radical. And that is part of this pathway of autophagocytosis, which Ralph Nixon had demonstrated many years prior to our work on oxidative stress, is one of the early changes of the disease in neurons that look relatively normal. Okay, so mitochondria, talking about biology 1 or biochemistry class, mitochondria are at the center of food metabolism in the body. Okay, and they have several characteristics. One is that they have a lot of metalloenzymes. The other one is they have their own DNA. It only codes for a few key mitochondrial enzymes. It's 16 kilobases in length, and it's organized much like bacterial DNA. We looked particularly to see whether there was an abnormality in the mitochondrial genome in case of Alzheimer's disease. So we looked for a 5 kb deletion, which we detected. But the more striking thing we found was that there was a tremendous increase in mitochondrial DNA between Alzheimer's and control, about a 3 to 4-fold increase, which was paradoxical because prior studies had said that there, if anything, there's a reduction in mitochondrial activity during the disease. And we confirmed that in our studies. But we found similarly mitochondrial proteins were increased, like cytochrome oxidase was also increased 3 to 4-fold. And so with that paradox in mind, we decided to look to see, are there really more mitochondria during the disease, even if they're not as active. So we looked at biopsy samples that were collected for diagnostic purposes, so it's early in the course of the disease, and classified the mitochondria in thousands of micrographs, and found, if anything, there is a decrease in mitochondria that have, well-developed cristae, double membranes, et cetera. Not a real big change, broken ones. Those are fixation artifacts. People describe those as if they existed in vivo, and I doubt any of them exist in vivo, and there's no difference between them. And then I'll come back to why I mentioned about the vacuolization, which is increased in Alzheimer's disease. And that is that when we looked at the in situ hybridization that I showed before of the mitochondrial DNA, you can easily do that with gold particles, nanoparticles, and you can know where the DNA is. And well, some of the DNA is in mitochondria, as expected. In the publication, you can see it there. The dominant place you see mitochondrial DNA within the neurons is in autophagosomes, these vesicular structure, as well as in the cytoplasm. In fact, if you look for cytochrome C oxidase, it's almost totally in the cytoplasm. So while the mitochondria have those enzymes, the dominant place is they're inappropriately localized. Okay, so having this finding, we decided to look at what is happening with mitochondrial dynamics. We looked at 3 different aspects. I'm only going to present 2. We looked at turnover, and there are alterations about their turnover, but we also looked at their movement by microtubules. That's the lower part. All of the mitochondria move on little tracks with a motor protein. Or we also looked at mitochondrial dynamics by fusion fission. So cells, when you say this cell has 1 mitochondria or 1,000, it still could be the same mitochondrial mass because it could be a thread-like structure throughout the whole cell, or it could be a lot of small things. So we counted the number of microtubules that were present in cells, and surprisingly, this had not been done prior to our study. People always talk about tau abnormalities and affecting microtubules, but nobody counted the microtubules. It's not that difficult once you have thousands of micrographs. So anyway, neurons that have, you can see in the very bottom, that's a micrograph showing a neurofibrillary tangle full of paired helical filaments, and microtubules coexist with it. So the 2 things can be together. And we counted either the length of microtubules or the number of cross-sections we would see. And then we looked at whether there was a difference. And what we found was there was a big difference in the number of microtubules in control people versus Alzheimer people. There was no difference whether the neurons contained paired helical filaments. Okay, and there's an internal control for fixation because you can look at smaller neurons that are less affected, and there's no difference. If you want to look at the upper right, there's an age-related decrement too, but it's more pronounced. Okay, but we wanted to be able to model this in a cell model, and changing microtubules with nicotosol or other things did not really replicate what we were seeing. So while the microtubules are changed, and certainly it's related to stability of tubulin, the tubulin protein also changes, and it's more of a stable form that I'm not presenting now, is that we also looked at some fusion fission, the thing I talked about that regulates the network of mitochondria. And this is regulated by a number of proteins, either that stick together mitochondria or pull them apart. And I'm going to focus on one protein for simplicity. You'll see the others there with data, and this is dynamin-like protein or dynamin-related protein 1, and it's the protein that squeezes mitochondria apart. So why do you care if you have one mitochondria or 1,000? There's a lot of reasons. Mitochondria need to be extremely dynamic, especially for a neuron. You know, they have these long processes. They need to move the material down. If there's one mitochondria, how are you going to move it down an axon? So one of the things is that mitochondria, by moving together, is the mitochondria DNA can develop problems, mutations, or other things. It's thought to be much more susceptible to oxidative damage or mutagenic changes, and it can be corrected by fusion there. Or you can end out like the small guys that are shown when you have the thousands, that you can turn over the deficient ones and just pull them out. Whether you want to have 1,000 or 1, you probably want to have it changing all the time. So we looked at these proteins, and you can see in the upper blots, dynamin-like protein 1 is vastly reduced in Alzheimer's disease and control. It's about a 75% reduction. That same protein is also present, when you look by staining, in the large pyramidal neurons. We had the real advantage, or something, again, I was quite surprised, if you look at fibroblasts derived from Alzheimer patients that are in the NIH repository, we found some of the same changes we found in the cells of the brain. The dynamin-like protein is reduced in Alzheimer's disease. That same protein is also present when you look by staining. And if you look at how mitochondria are distributed, it's not 100% like it was in the brain, but a high percentage of the cells from patients with Alzheimer's disease derived from them have collapsed mitochondria, like shown in that piece where the mitochondria are paranuclear. And what's important to note is that, these are fibroblasts, so they're vital stains and living cells that are being analyzed here. Okay, when you modulate the level of dynamin-like 1 protein by inhibiting it with a dominant negative or inhibitory RNA, you actually recapitulate the same thing. So dynamin-like 1 protein knockdown causes an Alzheimer-like change, and overexpressing it has a correction. You can see the same changes we've done in fibroblasts, derived from the fibroblasts converted to neurons, but I won't present that now. So what are some of the factors important in causing this change? Why is there a change between Alzheimer and control? Well, amyloid appears to be really a critical piece. It's not the only one. Is that if you take the cells and you transfect them with wild-type APP, you end up with more of this collapsed morphology. And if you put an inhibitor, APP plus the base inhibitor, you can reverse that. If you have ones with APP with a Swedish mutant, so it produces more amyloid, it also causes even more of the collapsed morphology and blocked by the inhibitor. If you do correlational analysis, you see the same thing with R-squareds over about 0.95. That if you measure the amount of amyloid and the amount of collapsed, and then we can actually measure how the dynamics by doing bleaching reactions. Okay, so this part of my talk shows the following. In normal, the upper neuron with its target, mitochondria are distributed throughout the cell where they're needed. In case of Alzheimer's disease, many of the mitochondria are clustered around the nucleus. There actually are bigger distances between the mitochondria. I left those slides out. We've seen that in culture, and we've seen it in the biopsy sample in the axonal hillock. There's a longer distance between each mitochondria. So they're poorly distributed, and we think that probably plays an important role in synaptic abnormalities and neuronal functional abnormalities. Okay, all of this sounds like I'm really a believer of the amyloid cascade because this is how it's one of the ways in which it's working. Well, at the same time we did those experiments, we did these looking at the quantitation of oxidative damage and amyloid just to document what we thought would be the case, that the two things cause each other. Well, that's not what we found. When we looked here, and this is a case of Down syndrome, and I'll present Down syndrome and sporadic Alzheimer's disease here. Down syndrome, the neurons are stained with blue for RNA oxidation, and the brown is amyloid. And you can see here the person with Down syndrome that's 17 years old has prominent oxidative damage, and the person who's 61 years old, which is advanced age for somebody with Down syndrome because of premature aging changes, you can see that the neurons are barely stained. Okay, when we quantitate those two things and divide it up into quartile decades, we find that the oxidative damage increases at least a decade before amyloid does. So it precedes it, and it's sort of going down. I'm not showing that in the upper panel, but if you look at those cases with amyloid and the amount of oxidative damage in them, you see a strong inverse relationship. We also looked at the relationship between neurons that had neurofibrillary tangles. So all of this oxidation is measured on a per cell basis. It's not related to neuronal loss. We look at each cell individually. And we found the same thing with neurofibrillary tangles, as I showed the picture before, of a decrease of about 50%. Now, why would there be less with neurons that have neurofibrillary tangles? Well, neurofibrillary tangles are associated with an antioxidant enzyme called heme oxygenase. And panel A is the hippocampus stained for heme oxygenase of a person with Alzheimer's disease. B is somebody without Alzheimer's disease. And you can see here more clearly in C, higher magnification. Heme oxygenase is an enzyme that takes heme, which is a pro-oxidant, and a product of metabolism of some of the enzymes from mitochondria, and turns it into biliverdin and eventually bilirubin that will be antioxidants. So it turns an oxidant into an antioxidant. So in the case of sporadic Alzheimer's disease, we wanted to understand, is the same thing happening as oxidative damage really early? Does it precede other changes? To address this, we looked at oxidative damage in incident cases working together with WashU, who followed normal people and their conversion. And those cases, we could see that MCI actually has very high level of oxidative damage, possibly higher than other stages. These are cases that we had that were entered a normal university center on aging. And again, we see the inverse relationship, which is highly statistically significant, and of amyloid burden going down. And if you look at this at duration of disease, it's the same thing. It's going down as the disease progresses. So oxidative damage is high early during the transition from normal to Alzheimer's disease, and then proceeds to go down. Same thing is true for familial Alzheimer's disease, diffuse Lewy body disease. And for those that are amyloid aficionados, I could have shown that it's always related to the amyloid 42 if you do the analysis. The 40 doesn't show this inverse relationship. And in this particular case, we had, let's see, I think it's, okay, there. We had one individual that was pre-symptomatic, had limited amount of amyloid in their brain. In fact, essentially was normal. They died of a heart attack, and they had higher than average oxidation. And why there's no scale bar, it's an N of 1. But I'm glad that people presented N of 1 studies of case studies. It's a rare case. Okay, so the same thing happens in all these cases. So why, so I mentioned about it for tangles, but why is this the case for amyloid? Why do we see this decrease? One of the things is, as I mentioned about the Fenton chemistry and plaques, the work of Ashley Bush and ourselves, which showed that they had metals. So we really wanted to understand how the metals were organized in real plaques. So we isolated plaques as an A, stained with antibodies to amyloid, B, which is showing that they're amyloid plaques, and I'll show EM of them later. And we did Raman spectroscopy, which showed that they had copper and zinc binding, those little bitty peaks, not too impressive, that are pointed out with the arrow. Okay, no indications of iron, okay? So why is that important? Well, when we looked at amyloid beta's effect on copper oxidation, if you place copper together with lipids or with vitamin C, it will auto-oxidize those. It'll catalyze their oxidation all by itself. And, but if you add amyloid beta, and that's probably to simplify, let's look at the 1-to-1 ratio, because that's what this is. It's a mixture of them. And you can see when you have copper together with one atom, one molecule of amyloid, it's essentially blocked. So we wanted to understand this even better, because I began to think that I didn't like the mechanism we proposed there, because it's a stoichiometric mechanism. One molecule of amyloid blocks one molecule of copper. I was convinced that there was probably a ferro-oxidase interaction, and that was what was going on. So we undertook to understand how the iron was organized and how each of the metals was organized within a plaque. So in this case, in C, you'll see an scanning electron micrograph of an isolated plaque, which happens, this particular one happens to have a blood vessel, most don't. And we did electron microscopy of them with an atomic resolution microscope. This microscope has a resolution of 0.6 angstroms. Well, and this particular specimen is different than one you probably have ever seen before, because the sample has never been treated with any metals. No osmium, no uranium, no lead. So the electron density here is from the specimen. And you can see the arrows point to little dots that are present within the amyloid deposits, those filaments, the wisps. And when we look at those little dots, they correspond to the crystal structure of magnetite, magnetic iron particles. And we actually demonstrated by a couple different methods, this particular method is holography, that they had magnetic properties, superparamagnetic properties. Which superparamagnetic, which is a fancy term for meaning that they don't have magnetism unless they're in a magnetic field. And for magnetite, it's not a trivial thing because it depends on the size. These are extremely small, and they're at a size that even could be non-magnetic. Okay. In the more recent study, using an X-ray microscope, and an X-ray microscope is not as simple as some X-ray that you take of a person. It's hooked to a synchrotron at Oxford for high-intensity light, and it allows you to directly determine not just the metals, but their redox state. And that's critical to know whether they could catalyze redox reactions. In particular, we wanted to know whether there was copper 1, copper 2 redox cycling. And that you can't do by any of the methods we had available to us. So that's why we collaborated with them. When we did this, we also analyzed iron. And we found something very surprising, okay? So we found copper 1 and copper 2, which shows that the copper that's in plaque is able to do redox reactions and probably is responsible for the early 1997 paper of changing iron redox state, because that's ferroxidase activity. But in addition, we found that there was an abundance of copper 0 and iron 0. Neither, to our knowledge, has ever been described in the human body. Copper 0 has been described in biology from bacteria. And to our knowledge, iron 0 has never been described in biology. Why? It's extremely unstable. I mean, all you have to think the rusty nail idea, except that this is individual atoms of iron that have that. So we still don't know how it is formed or how it is stabilized. Is it there because mitochondria are something like bacteria and are being able to generate it, or was there bacteria within the plaque? Those are all things. And whatever it is, it's stabilized there because the plaques are stored in an air environment for long time periods. We don't take any particular caution. Okay, so these are all different, showing the metals that were there. In addition, we did further confirmation about the magnetic state of the iron. Okay, so how is this metal organized there? We still don't completely know. I thought it would be related to histidines bound within each amyloid, because that's what you see when you look at amyloid fibers, amyloid synthetic. But it's more complicated than that. And that is, if we take a plaque core, and this is just a different type of electron microscopy, still scanning, in the upper left, and we remove the copper, you see the plaque falls apart. But it stays together as filaments. We know it's thioflavin positive, but right now am trying to define whether the beta sheets are maintained. All of this suggests to me that the binding sites are actually between the fibers. But I would say that a lot more work has to be done there. Another thing we found, very surprising, is if you took amyloid beta and you put it in the appropriate buffers and added copper to it, you actually ended up creating something in vitro that looked an awful lot like a plaque, 10-micron balls that are thioflavin positive. Okay, and then mechanism for reduction. Does amyloid beta play a role in this? Well, amyloid beta, in this particular advanced mass spectrometry analysis, was able to reduce copper. And that's what's shown here. Okay, so this would be, we've actually found pretty good evidence for Fenton chemistry actually playing a central role. And where does this lead in terms of a treatment or anything else? Well, this is a picture that Mark Smith and Mark Avinovich came up with years ago. And the idea that amyloid and tau, instead of being the culprit in the disease, are critical protective responses. And the neurons are maybe not in a vacation, but are resisting death. And because we were very much trying to understand how do neurons persist in Alzheimer's disease. You know, those neurons I showed have tangles for years. They don't just drop out. What is the basis for that? So I talked here about a bunch of these things here, the abnormalities of Alzheimer's disease, amyloid deposition, tau deposition, oxidative damage, antioxidant responses, and I could have talked about more classic antioxidant responses because they're both in signaling and antioxidants are increased, vascular degeneration, blah, blah, blah. Those are just the ones we studied. So if you add together the other ones, you have, a list of a couple hundred things. But yet, with all that screw-ups, the neurons managed to live. I mean, Paul Coleman estimated 20 years, but certainly years. And so this is another part of the hypothesis, and I think fitting along with people we're talking about, do the numerous abnormalities mark a new homeostatic balance that we call Alzheimer's disease? It's stable, it's not functional. Is it similar to hibernation, which is stable and not functional, right? Organisms, like we studied Arctic squirrels in Alaska, and they lower their metabolism almost to zero, and their brain is refractory to damage. And if this is the case, then what are you doing when you target any of these changes? You just destabilize you destabilize the compensations. You're not benefiting anybody, and you get what you get for the amyloid treatments. For the most part, people get worse. Okay, so in summary, what I presented is oxidative damage is an early feature. I always emphasize that because every time I see the amyloid cascade, oxidative damage is in the in the basement, okay? It's always at the bottom like it's a late change. It is not a late change. It's either coincident with the other pathology or precedes it. And why I think in Down syndrome is there's probably oxidative damage plays a more primary role. In Alzheimer's disease, it plays a very coincident role. Mitochondrial abnormalities are critical to them. They occur in the same cellular location. They're probably important in generating the metals that are there. And it's important to know for the metals, when I talk about it, isn't as simple as chelating therapy. Those metals are probably being redirected to having a protective function also. Okay, and then amyloid is linked to mitochondrial turnover. That's from our studies, but there are also other studies that amyloid toxicity requires functional mitochondria. Let's see, copper and iron abnormalities are intimately associated both with plaques and tangles. Our preliminary analysis of metals in tangles, which we're still in the process of finalizing, is that they don't have the same metals. They have both copper and iron, but they're not metallic. And lastly, a cure, whatever a cure means, and I think a cure comes closer to what has been shown today, that people actually get better. A change of the slope of getting bad, I don't consider to be a cure. I mean, it'd be nice to think of people being totally reversed, which Dale maybe has one, but if people got better. That's certainly close to if it was your family member, you would want them to be treated that way, not noticing 22% change. It requires understanding and targeting the complex compensatory abnormalities that are going there. You can't just wholeheartedly take them out and say, I'll remove the amyloid, I'll remove the tau, I will change the blood vessels, and end up with more cerebral bleeding. Okay, so that's my talk, and it predicts that continued efforts on treating amyloid, which I think will continue, no matter how many articles I write about how silly it is. I've ran out of new arguments. I mean, the last one I had, which I think I haven't written enough about, but I did debate and won the debate, was about genetics, about the whole issue of genetic causality is misunderstood. Because that's the main argument, is the genetics. And all it is statistics. And you know about what statistics shows. Okay, so I'm available for any. Do I have time for questions or no? One question. Okay, thank you very much. Okay, no questions. Okay, perfect. Oh, yeah, I should have.
So in terms of the expectation that everyone is protected from the at-home trials and the other trials that are ongoing with the vaccines that actually lower the amount of amyloid, the claim that they're making is that there's a 22% reduction in the rate of progression. But doesn't your picture of this indicate that they might have more oxidative damage?
Yeah, I haven't analyzed any of those cases, number one. Number two, most of the people got worse in that trial. That was when they did a subgroup analysis.
So, okay, so, and they still managed to get...
If you do enough, yeah. If you do enough people, there was 3,000 people in that trial. How many people constituted? It was really a small number, correct, Dale? I don't know the number. It was an extremely small subgroup that was analyzed. It might have been like 30 or 40.
One dose in one trial only.
One dose in one trial, and not the whole range of patients, even in that group. If you're dealing with the statistics, and there's debate about whether it removed all the amyloid also, but I'm not an expert on that. I can tell you there's been several commentaries in CHAD in which people from your institution, actually, from UCLA, have, in fact, both your institutions, that's right, have argued that the imaging techniques are not specific. But I'm not an expert. I actually decided not to get involved in it, except for allowing it to be published. Okay, thank you.
Okay, I want to welcome our next keynote, and that's Deborah Zelinsky. And Deborah is the director of the Mind Eye Institute. She's an optometrist and working on very innovative ways of integrating optometry and manipulation of visual stimuli to improve neurological disorders. So, Deborah?
Thank you, Chris. I'm not tall enough to see the monitor down there, so let me see. Is there a way to move the monitor? Or I can move me. How's that? There we go. Thank you very much. As a clinician and a researcher, what I'm here to do is to explain why eye care policy should include brain mapping and retinal neuromodulation in order to help patients better. There is a slide clicker. There, got it. There we go. I want to thank Babak Kateb for allowing me this opportunity. I want to thank the Mind Eye Institute staff for helping support this venture, and the entire brain mapping organization for promoting collaboration between different types of professionals, and all of you for listening, especially now that we're running so late. 2020 is considered perfect eyesight, and it has been since 1862 all the way to the present day. I started school 40 years ago, and I learned that light bounces off of targets and it's dispersed on the lining of the eye called the retina. And we learned that being able to read 20/20 on the charts means that you don't need any glasses. That type of testing shows that light came into the eye and was an input mechanism. But in reality, with new scientific discoveries, they're finding it's a two-way street. It's not just inputs. There was a book written called The Retina by John Dowling in 1987. It's a huge book that covered retinal processing in all different animals, including humans. 25 years later, in 2012, Dr. Dowling wrote a second edition, And in the preface, he wrote, "I didn't realize that in 25 years there'd be so many new things about the retina that it would take another book." He was shocked because when he finished in 1987, he thought everything was covered. But in 2002, new cells were discovered that linked with hypothalamus functions. And now we're 10 years after his 2012 second edition, and they found a new cell about 4 months ago in the retina because instead of microscopes, they're now using nanoscopes. And these cells are so tiny, nobody knew they existed, and now nobody knows what they're for. They're shaped like a handbell, and they're called campana cells, which is the word for bell in Italian. But the retina keeps having new discoveries, and they find more and more linkages into brain function. So if you take one point home from this speech today, it's that 20/20 eyesight is something that's slow, and it's at a conscious level. The signals that are used to identify details clearly. Because it's slow, it means that we don't use it all the time. We use it when we choose to put attention on a detail. Here's an example of what happens through a closed eyelid. We were expecting that if you close your eyes, there isn't any eyesight and there's no reaction. You should be able to put anything in front of your eyes, and if the eyes are closed, nothing will happen. But since light still gets through a closed eyelid, here's what happens to sound perception.
So I want you to close your eyes, and wherever you hear the bell, touch it with one finger. Ready? Touch it, one finger.
Now he's not able to visualize
Now we're gonna try
the
a different
space
tone.
around him correctly.
It is.
But this bell, he can because the
So
frequency
we're
is
going to show you
different.
that on the other side. Find it here.
He can't find the lower pitch, but he can find the
and
higher
find it with
pitch.
this tone. Does it differently.
Now, what happens here is that I put blue glasses on his face. So he has his eyes closed. The blue is a high frequency as opposed to red. And what it's doing is filtering only blue light getting into his brain. The reason I say brain is because the retina is a piece of the brain. And the blue light is going through his eyelids, and now he's able to find that low
the
pitch.
blue color makes a big difference because it's allowing the higher frequency sounds in and getting rid of the low frequency sounds.
Okay. The point of that is that, oops, how do I go to the next? There we go. The point of that is that light traveling through a closed eyelid is affecting spatial perception, and it's affecting brain processing. Retinal stimulation affects systems that are completely separate from eyesight. They're called non-image forming pathways, and they link directly with brain processing. So here's an example. You have image forming sections in your brain and non-image forming sections in your brain. The retina sends signals through the optic nerve to both both places. The image forming is central eyesight when you identify details, and peripheral awareness, which is awareness of the background. Now, some people think of periphery as way on the sides, but it's not necessarily. If you're looking at somebody's nose, then their earring might be in your peripheral eyesight. Peripheral really means whatever you're not paying direct conscious attention to. So if you're reading a book and you're looking at one word on the page, your peripheral eyesight is governing where the end of the line is and where the beginning of the next line is. So it's not way on the sides necessarily. Then you have these non-eyesight forming signals, and they're linked with posture mechanisms, metabolism mechanisms for sleep, mood regulation, and your fight or flight nervous system. The sympathetic nervous system is very much entwined with your retinal stimulation. Just think of what happens if you're driving and you see a police car chasing you all of a sudden. Your fight or flight turns on instantly, and your heart rate goes up. The peripheral retina, which is different from the central retina, has both non-image forming as well as image forming signals. The image-forming signals are used to determine speed, location, size, and shape, and we use them all the time. For instance, in driving, the only time we use central eyesight in driving is to look at a license plate or a street sign or to find an address. Most of the time, driving requires peripheral eyesight. And the same with reading. If you're skimming and just scanning along on a book, you're using peripheral eyesight to judge the shape of words. You use your central eyesight when you're trying to proofread and look at commas or periods or small letter changes. 20/20 eyesight isn't going to save this man. The key point I want you to take home is that 20/20 eyesight is slow and it's processed at a conscious level. So his non-eyesight, which are the fastest signals, those are involved with his posture, metabolism, and mood, as I just said. And his peripheral eyesight is busy looking at the speed of that bear, the location, the size, the shape. He doesn't care whether it's a bear or a gorilla. He doesn't care what color the bear's eyes are or how many teeth it has. That's what you'd use the 20/20 eyesight for, to zoom in on identification of details. He's busy in survival mode, and it doesn't matter about the details. It matters about the concept. And his peripheral eyesight also has to keep him from falling off the cliff. So peripheral eyesight does a lot. Now, emotions can override the 20/20 eyesight. For instance, I once heard a noise in my basement, and I opened the basement door and saw something in there that didn't belong in my basement. It was moving around, and I got scared, slammed the door, and called the animal control people to come and get it out. And they came and they said, well, what is it? Is it a bird or is it an animal? And I said, well, I don't know. And they said, well, it's easy to tell. One has fur and one has feathers. And I said, but I don't know which one it had. And then they said, well, did one have a beak or did it have a tail? And I said, I don't know, I didn't see it. And I said, but I did know exactly where it was in my basement, how fast it was moving, how big it was, and what shape it was. And that's my peripheral eyesight. So when I opened the basement door, my peripheral eyesight glimpsed all of that in an instant. Because I was so scared, I shut the door so fast, I didn't choose to have my 20/20 eyesight point my eyes and then look at it and then zoom in my focus. So peripheral eyesight triggers a lot of brain activity and a lot of decision making way before 20/20 eyesight kicks in. And each person has a different reaction and response. So when we see patients and we're measuring their peripheral processing, then we look at what responses they have, and they're all individual. So when we talk about precision medicine and individualized treatment plans, you can do that from optometry also by stimulating peripheral signals and seeing how the responses are. So for instance, in the demonstration on the video before, when the person wasn't able to find a low-pitched bell, and he was with blue glasses, there are other patients who can't find the high-pitched bell, or other patients who can't find it just on one side, or other patients who need green or red, something different. Each patient is a nice puzzle. Oops, that's the wrong direction, this way. So this is why eyewitness accounts differ. You can have a car accident or a robbery, and everybody is uniquely paying attention to different things that interest them or that scare them. And the policeman can come and try to get reports, and they'll have 5 different versions depending on what each person is paying attention to. A key point is that peripheral stimulation is activating brain processing without central eyesight necessarily being involved. And the people are not always concerned about the same details. I had a patient once in my office, and they left, and my employee said, "Did you see what they were wearing?" And it was like, "No, but did you see how pretty their eyelashes were?" And she hadn't looked at their eyelashes, I hadn't looked at what they were wearing. Details are just whatever is individual to the person. Some people watch hairstyles, some people watch fashion, just different. There are some functions that are meant to be automatic, where they're running unconsciously. So for instance, we're not aware of swallowing until we're choking or having a sore throat. We're not aware of breathing till we have a stuffy nose. Can any of you tell me how many times you just breathed in the last minute? You're not paying attention to your breathing. It's just happening. And that's the neat thing about peripheral eyesight, that it's taken for granted all the time. We use it without ever thinking about it. If you're walking and you happen to hit a curb, you can be talking to somebody and your peripheral eyesight will see the curb and then make your step change. It's changing your posture and your movement so that you won't trip and fall. It'll see a pothole. It'll see something flying at your head. Peripheral eyesight is always taken for granted until you have an autonomic dysfunction or a brain injury. Then all of a sudden, it's no longer there. It's disrupted. It doesn't work right. So why do we even care about peripheral eyesight? Well, eye examines didn't emphasize them when the 20/20 was invented in 1862, but people then were looking at huge amounts of peripheral eyesight. Now we're using peripheral eyesight very minimally. There's a picture here of the subway.
To match the lenses in 1862. The instrument for looking at your eyesight to put in front of your face, that's called a phoropter, and it was from 1904. Now, as you see the iterations that go through 2021, the instrument is sleeker and fancier and now computerized, but it's still doing the same thing. It's still blocking off your eyesight. We're still measuring central eyesight, and we're still doing it with conscious attention. The sight test in the offices, it's meant at a conscious level, and it's designed specifically to find pathology. So it's looking for damage to peripheral eyesight. And they say, look straight ahead and push a button when you see something out of the corner of your eye, but you're consciously paying attention to where is the little dot coming from. So it's not the same as when you're walking around and not thinking of your peripheral eyesight.
So the central 20/20 eyesight, which I want you to go home realizing is slow and at a conscious level, it's part of the what do I see? It's an identification process. The peripheral eyesight, as I said, is used for pathology, but it's part of the where and when, the space and time judgments. and it's more of a navigational process. So you have identification and navigation. You know this from when you get new glasses. Sometimes they often feel funny, but the typical person can adjust to a prescription designed for the central eyesight. So the eye exam is fantastic for the bulk of the population, but an overwhelming percentage of brain injury patients, or people with autonomic dysfunctions, or people with even long COVID, those are people that have a lot more trouble adapting to changes because the peripheral retinal signals going in is confusing. So what I'm proposing, since the retina is part of the brain and made out of brain tissue, is to map the brain onto the retina, use brain mapping at the retinal level. There's a point-to-point correspondence with cortical sections. And if you did that, you could then use it to zero in on brain injury. I have a speech tomorrow on how you can use the retina as a poor man's MRI, and you can selectively activate different spots on the retina to route through various cortical pathways. It would facilitate and individualize rehabilitation. So why is retinal mapping an important part of brain mapping? Because it's non-invasive. The newest research shows that retinal degeneration is a reflection of what's happening parallel to brain degeneration. So there's studies that show that in multiple sclerosis, not only does the brain have signals that are not working right, but the retina is changing its thickness. In dementia, the retina gets thin, but always in the same spots. That's with Parkinson's and dementia. There's studies on schizophrenia, studies on bipolar disease, looking at retinal tissue. And now they're trying to figure out, is it the chicken or the egg? Does the retina degenerate first and then the brain, or does the brain degenerate before the retina? But it's an incredible avenue for research. And few people realize that eyeglasses are one way to directly alter brain activity, because the clarity of a target is just one aspect of visual processing. Visual processing is an enormous thing. So even when you have 20/20 eyesight, which is slow and at a conscious level, you might still need glasses for other reasons. We're not where we were 40 years ago when I learned that 20/20 meant you didn't need glasses. You could use them to help you listen better, like in the video. You could use them to feel more comfortable. It's just no longer acceptable to assess only central eyesight. The policy needs to be changed, and it's vital to adapt the eye exam for patients who have autonomic dysfunctions, attention disorders, brain injury, long COVID, and autism. Now, how can we change the policy for clinical practice to catch up with current science? There's a lot of ways. We can standardize the curriculum to emphasize the overlap between professions. We can use occupational therapy codes that are already existing, talking about sensory processing, to be used by eye care professionals. We can create new diagnosis codes to talk about these non-image forming disruptions. In glaucoma, for instance, one of the first bunch of cells that's damaged in the retina are the ones that are linked with sleep. So a lot of glaucoma patients have sleep problems because these particular cells are affected. There's also incorporation of peripheral awareness into the assessment in a standard eye exam. Up until now, people thought of neurology and cardiology and optometry as being very separate, but they're not, because when you change lenses in either optometry or ophthalmology, you're affecting the brain directly, and you're instantly triggering the central nervous system and getting reactions. They have to be thought of as together. So one way to do this is to start with collaboration. And I think that can begin with brain researchers needing to include the effects of eyeglass prescriptions on neuromodulation, and optometrists needing to learn more about these non-image forming pathways. Collaboration between the scientific community and rehab professionals and mental health care providers and legislatures has got to make a better quality of life for patients. It's going to save health care costs, and it's going to increase productivity, and it can take some people off of disability. We have so many patients on disability that, you know, they have mismatches between their auditory maps of the world and their visual maps, between their right side and their left side, between central and peripheral processing. And when we put specific types of glasses on them, all of a sudden their brains work differently. They can go back to work and be comfortable instead of uncomfortable. The Society for Brain Mapping and Therapeutics that all of you are a part of is a different kind of organization, and it's based on collaboration between professions. This is the wave of the future. It started with engineers, neurosurgeons, and scientists, and it's now expanded worldwide to include the military and different brain initiatives. And it's got a brain technology innovation park that's just on the cusp of happening, and that's going to be even more collaboration. It's going to allow a platform for eye care to be one of the first stops for brain function. When Babak made this organization and talked about breaking boundaries of science, he wasn't kidding. I started in this organization in 2009, and there were about 20 people. And now, before COVID, there were hundreds of people. And even now, there's about 800 or 900 speakers from all over the world in so many disciplines, and it's all under one umbrella of the brain and the spinal cord and the retina, because all of those together are the central nervous system. So we're here today, but where tomorrow? Today we know that retinal activity involves much more than 20/20 eyesight, because 20/20 eyesight is slow and conscious. But there's also these unintended consequences of glasses. For instance, there's a 1969 episode of Marcus Welby. For those of you who are too young to know who Marcus Welby is, he was the older version of House. There's a TV show called House, and where House figured out things that nobody else could figure out. And Marcus Welby was a similar TV show from earlier where he was a grandfatherly figure who always solved things. And the episode of Marcus Welby shows a lady saying to a man coming to Marcus Welby and saying, "Thank you, Dr. Welby. You showed me that if I got glasses, my digestive problems would go away." And in 1969, Marcus Welby knew that if you put glasses on somebody, their digestive system would change. But here we are, 60 years later, and 50 years later, and we don't know that, but it's true. And I had a patient who was a bird watcher, and she used to always have trouble figuring out where to find the birds. She could hear them, but she couldn't place where they were. She was like the patient I had where they couldn't find the bell. They heard it. And we put glasses on her, and she came back and she said, "Oh my goodness, now when I hear a bird, I can turn my binoculars immediately and see them because I know where the sound is." So the concept of where we are in the future is that visual stress induces physical reactions. So when we take away visual stress, we're going to be able to alter people's lives and help people in ways they've never realized before. Because people don't realize if they see 20/20 out of each eye, but one eye sees it easily and the other eye sees it with struggle, they're still graded as 20/20, but there's a circuitry imbalance. So the brain is under total stress. So the challenging patients require increased sophistication of testing and treatment. So I hope you're going away realizing that even if they're 20/20, they still might need glasses to function more comfortably and efficiently. So how are we able to help, and who can we help? We're able to help people with learning disabilities, mental illnesses, brain injury, PTSD for veterans, autism, genetic problems, anybody whose brain has not learned certain systems, that it hasn't developed properly, or somebody whose brain has been disrupted with the development. Trauma patients are like that for the PTSD. There's something that has disrupted the linkage between peripheral and central eyesight. So, for instance, in a veteran, their peripheral eyesight isn't taken for granted because it's always on hyper alert. It's always looking for a sniper, for somebody around. Peripheral processing is crucial, and yet we shouldn't any longer, for the brain-injured population, be blocking it off when we test them. So eye care provides this missing link by changing the brain through retinal neuromodulation. For instance, we know 85% of people with high blood pressure have no known cause. But changing light on the retina directly affects the central nervous system. So wouldn't it make sense to do some research on maybe eyeglasses can help the patients where blood pressure isn't regulated easily with medications? There also might be other systems that might be affected, such as digestion. So we have to leave 2020 in the 20th century because we're using testing that was designed in the 19th century, but we're currently in the 21st century. So here we are using 160-year-old testing that was brilliant at the time, but needs a change now. So I urge you to help us as the brain mapping group change policy. I want to thank you for your time, and if you have any questions, you can write me at info@mindeye.com. Any questions here? Thanks. Is there a next speaker that I can introduce? No. Well, thank you very much.